So welcome everyone to the 21st Ecological Integration Symposium. We are in the Carrion Ecology session. So we have 4 really great talks today. And our first talk is from Lauren Beebe, and she is presenting the extreme temperatures impacting the larval stadia of the hairy maggot blowfly, Chrysomya rufifacies, and the secondary screwworm, Cochliomyia, macellaria, in the Diptera, Calliphoridea.
All right, we good?
Yeah, we can see it.
Okay. Thank you all for being here this morning under these interesting circumstances. So I'll go off, talk about a tale of two flies. So the 2 species I'm going to be talking about today are Cochliomyia macellaria and Chrysomya rufifacies. Both are frequently found in Texas and are very important in the study of decomposition ecology and forensic entomology. Macellaria is native to North America and spans from South America up into Canada. It's theorized that it is a thermal generalist because it's in its native range, it's exposed to such a wide variety of temperatures from extremely cold to extremely warm. Rufifacies is not native to North America. It originates from the Australasian regions of the Eastern Hemisphere and is theorized to be a thermal specialist because temperatures in its native range don't fluctuate as much, and they tend to be much warmer in general. So it has made its way to North America, and as I said before, they're often seen together here in Texas. So this is a typical curve for what a thermal generalist might look like. On the y-axis, we have phenotype, which is any measurable characteristic on and body temperature on the x-axis. So a generalist performs fairly well throughout a wide range of temperatures, compared to, say, a cold specialist or a heat specialist that would outcompete the generalist in their optimum temperatures. So the theorized heat specialist rufifacies is predatory and will feed on other maggots. It has displaced other species after invading, but not macellaria. So why is that? What we know so far is that there are records of macellaria being outcompeted when both species are present on the carcass. And when rufifacies numbers were reduced, macellaria is much more successful. So it was predicted right after their invasion that there would be a reduction of macellaria across North America 30 years later, and they are still here. So macellaria has a higher survivorship when colonizing more than 2 days before or after rufifacies. And rufifacies has extremely low performance when colonizing 4 days ahead of macellaria. So facultative predation of macellaria by rufifacies was frequently observed in the lab, but not in the field by Pimsler et al. In 2019. And segregation was observed between the species in the field. So rufifacies was in its own little bubble, kind of on its own at the soil body interface. So the questions we had were, could temperature play a role in these species' interactions and coexistence? Do they have different responses to temperature, different limits? So I decided to test both species' thermal tolerance of high and low temperatures using the ramping method. So I'm going to focus on the responses of the larvae, because this is a stage where most of the competition occurs between these species, feeding in masses on a carcass outside. So the measure of an organism's thermal tolerance helps estimate a survivable range of temperatures. Extreme temperatures can be lethal on both ends, and for insects, development is restricted within this range and regulated by temperature. So this is a thermal image of a cadaver in Texas, and as you can see by the range of temperatures on the right-hand side, it gets to almost 65 degrees Celsius, which is extremely hot. If you can see my mouse, the area, the circular area to the center left of the image is the in the abdomen, and this is just a pile of dead maggots, and which is makes sense because the edge of this ring is bright white, which is about 65 degrees. And this picture was taken in May in the morning, so this isn't even peak temperature or during the one of the hottest months in Texas. So this is what a typical performance curve would look like, with performance, any measurable characteristic on the y-axis against body temperature on the x-axis. So we'll see an increase in performance with body temperature up until an optimal temperature, after which point performance severely drops off if temperature is raised at all. So at the intersections, we have critical thermal minimum and critical thermal maximum. Both of these are measured by knockdown, which usually is the loss of the ability to locomote. So initially, because Texas is still hot and we have been observing dead maggot masses in the field, we were focused on the right side of this curve, especially because a change in temperature has much more severe consequences. But we also want to look at the left side of the curve, especially because the invasive rufifacies has been limited by cold temperatures in its range expansion and is most frequently found in the warmer southern states, but is known to move northward during the summer months. So this is a graph of the total occurrence records of the adults of rufifacies and macellaria collected by month in Texas. So rufifacies is the dashed line on the bottom. So as you can see, they start to pick up in the summer and really peak early fall, and then once it starts to get cold, their numbers plummet really quickly, as opposed to macellaria, which is common throughout the year. So objective 1 was to determine the critical thermal maxima for these species. So again, this is measured by knockdown, which is basically when the organism stops moving. So an organism will be exposed to steadily increasing temperatures over time until knockdown is observed, at which point that is that organism's CT max. So it is possible to recover after being knocked down at high temperatures. If you're removed from the stressful environment, you could recover and continue on with life. So for one generation of flies, I took 40 individuals from each instar, the first, second, feeding third, and post-feeding third instars. This was repeated for 2 generations in both species of flies. So I used this aluminum plate set in a hot water bath connected to a data logger and a control plate. So the larvae were set in here and observed while the temperature increased until knockdown was reached, at which point they were immediately taken off. So this is the results for that. Temperature on the y-axis, stadium on the x, and rufifacies is in red, macellaria in blue. And as you can see, the first instar larvae are much more susceptible to higher temperatures than the later stages. Kind of tapers off at both the feeding and post-feeding third instar. And if you look, rufifacies in red compared to the blue knocks down at slightly higher temperatures, and that was pretty consistent throughout the whole thing. So a generalized linear mixed effects model showed that stage had the largest effect on the data. No significant difference within stage, but there was a species effect across stages. And if you remember back to the thermal image, the range of temperatures, 33 to 65, if you look at this graph, is stressful for that whole image. Anywhere on it would be stressful for a maggot. So this is the survival after 24 hours being exposed to the heat. So percent survival on the Y and stadium again on the X. As expected, the older larvae had much higher rates of survival than the younger. Rufifacies in red, macellaria in blue again. But this time, macellaria tended to survive better than rufifacies, except for this feeding third instar. And ANOVA showed that both species and stage were significant factors here. And this is the control. So this was expected. really high rates of survival across instars. ANOVA showed species to be a significant factor, but not stage, and these results were significantly different from the treatment. The second objective was to determine the critical thermal minima. So instead of increasing temperatures, it's a drop in temperature, so steadily over time. So again, for one generation, 40 individuals from each instar were taken. However, due to this virus, we have been unable to collect data for the last 2 rounds within separate generations, and unfortunately, only one species was able to be documented. I was hoping to have a lot more data for this presentation. So same plate was used instead of a hot water bath this time. It was an ice bath. So again, larvae were placed on there until knockdown at their CT min was reached, and then they were immediately taken off. So these results are just for macellaria, and this is 1 round. So we have temperature on the y-axis, instar on the, or stadium on the x, and this looks a little different from the last set of results. So first instars are a little more sensitive, knocking down starting around like 13, but the cutoff pretty much for everybody is 4 degrees Celsius, except for the feeding thirds, which might be fixed by some more rounds of knockdown. But so then if you compare this to the critical maxima knockdown, if you remember, the range of temperatures on this for the knockdown was about 30 degrees compared to just 10 degrees for the minimum. So the spread of knockdown is much smaller for the colder temperatures. All right, so for a second, we're going to focus on the post-feeding third instar results for macellaria, because I actually did manage, while finalizing methods, to run a preliminary study of rufifacies in the post-feeding third instar, so I at least have that to compare. So the circled one is still macellaria in blue and rufifacies in red. And as you can see, rufifacies was much more sensitive to the cold than macellaria was, knocking down about 6 to 7 degrees higher than macellaria. And this is just the post-feeding third instar, so I really don't know what happens for the rest of them, but that's a pretty big difference. So in conclusion, species and stage were significant factors in response to high temperature. Rufifacies knocks down at slightly higher temperatures, but macellaria has slightly higher rates of survival for CT max. And stage at this point does not seem as great a factor, contributing to critical thermal minimum. And with the limited data I do have for rufifacies, it does look like species may be a significant factor in their lower knockdowns. So from our questions earlier, yes, they have different responses to temperature, different limits. So I think it's safe to say that temperature could play a role in their interactions and coexistence. So they could be inhabiting separate thermal zones on a body. Again, the thermal landscape of a body or a carcass outside could span 30 degrees, it could be 50, it could pretty much be anything. So there are lots of spaces for the different species to inhabit if they so wished. And macellaria could be colonizing earlier to develop more thermally tolerant stages, to heat out competitors or predators in their large maggot masses. They do produce a significant amount of heat. And macellaria could be increasing their populations during the cooler months when rufifacies isn't suited to develop. So these could all be reasons why macellaria is still here. And then our last question was if both species fit their theorized generalist and specialist roles. So if we put the 2 species in their theorized curves, actually setting their average knockdowns, at least for the post-feeding third instar, the higher ones at the top, lower knockdowns on the bottom. Obviously the scale is not quite right, but it seems to fit these curves fairly well, at least for their post-feeding third knockdowns. But if we remember, rufifacies didn't survive as well as macellaria at their upper knockdown, so it could be that the macellaria's curve is shifted slightly to the right. So do they fit their theorized roles? So far, with the information that we have, I believe they do. And with that, I would like to thank the Tyrone Lab, Dr. Travis Brush, Jeffrey Young, Dr. Pierre Lin, and the Tomberlin facility, and everybody at WISE. And also my family in Indy, who is actually, I think, seeing me present for the first time ever. So thank you, and I guess I'll take any questions.
Thanks, Lauren. That was really great. I'll let anyone else ask questions first, but I do have a question for you. But I'll see if anyone else has a question first. Okay. So on my rats, I see Chrysomyia rufifacies and Luciliaxemia.
Mm-hmm.
So we're starting to do, Travis and I are starting to do some thermal work with both of them. And it seems that eczemia can tolerate really high temperatures. So they seem like they could also be a heat specialist. But we have these white and brown rats, and on whatever rat we're looking at, the only time we see both rufifacies and eczemia is on white rats. So we don't know if that's a temperature thing, or there also is fire ants present during that time. So we don't know if it's fire ants or temperature. But do you have any ideas about that?
I mean, if they're white, it could be the absorption of sunlight on the darker rats that could play a role in the temperature difference, or
yeah, and that's what, to me, I don't know. Well, that's what the thought is, that since they're out in open environments, because we see even with fire ant activity, they're not on the brown rats. So we wonder if it is the solar radiation increasing the temperature.
And that could be deterring the ants from the brown rats, or
yeah,
whatever.
that's I don't know the answer to it. I just wanted to know what your thoughts are, because you do the thermal work with both macellaria and rufifacies.
And do they come from the same supplier?
Yeah.
Okay. Then, yeah, I don't know.
Yeah, it's really interesting. But that's what I was thinking before watching your talk between this one and your defense. I was thinking rufifacies was more of a generalist, just because eczemia seemed to outcompete them in the heat realm. So I don't know. I just wanted to see your opinion.
Yeah, and it could be that, I mean, because macellaria was also in the other study with the static temperature, so putting them in incubators for, like, at stressful temperatures for longer periods of time, so like setting them in at 45 degrees for an hour or two, macellaria was surviving a lot better than rufifacies was. So it could be that rufifacies, kind of like mid to high temperature specialists, since they don't perform well in the cold.
Yeah. Okay. So yeah, they're certainly not cold.
Yeah.
I don't. Well, thank you. If anyone else has any other questions, we can take those.
You can also type them in the chat if you don't want to.
And I can read the chat if anyone has any questions to type.
If not, that's fine too.
I was going to say, we'll give it a minute. And then I'm going to give hostship to Jeffrey so you could start sharing the screen. But if anyone has any questions, we can still answer those. All right. So it's 10:20. You can access Lauren's email through our program book. So if you do have any questions that come up a little later, or anyone watching this on recording after the fact, you can still contact Lauren and ask for questions. So you can check out our program book through our website to do that. Thank you, Lauren.
Yeah, thank you.
So our next speaker is Jeffrey Young, and he is presenting the impact of injury and heat shock on the life history traits of Cochliomyia macellaria and its implications in forensic entomology.
All right, thanks, Sam. So just before we get started, I just want to put a warning out there that I do have a few dead animal pictures. So decomposition is a process where non-living organic matter is broken down and recycled back into the ecosystem. In comparison to autotrophic biomass, though, heterotrophic biomass, so carrion, makes up a very small amount of the total biomass decomposition total biomass that undergoes decomposition. But carrion is still an important source of nutrients, and it's going to be the focus of my talk today. So carrion is any non-living heterotrophic organism, and it can provide a pulse of carbon, nitrogen, and other nutrients to its immediate environment. And carrion decomposition has traditionally been described in stages, and Payne described 6 of these stages: fresh, bloat, active decay, advanced decay, dry, and remains. And nowadays, though, decomposition is more recognized as a continuous process, but stages of carrion decomposition is still widely used in the literature, mostly just to help with understanding the process itself. So the organisms that I'm interested in, specifically microbes and blowflies, are more active during the earlier stages of decomposition, specifically fresh, bloat, and active decay. So microbes, almost immediately after death, start breaking down the remains, and the larvae of blowflies feed on the soft tissue, as seen in this picture here. You can see the maggot masses. So as I alluded to, there are several organisms involving carrion decomposition, anything from microbes, invertebrates to vertebrate scavengers. And more recent studies have looked at the interactions between them and have recognized the importance of these interactions with respect to carrion decomposition. And as I mentioned, I'm going to be focusing on microbes and blowflies. So with blowflies, the adults will go to the remains shortly after death. The females will lay eggs, and larvae will hatch from these eggs and go through 3 larval instars while feeding on the soft tissue. The last third instar will leave the body once they've fed enough or there's not enough food, and they will pupariate away from the body. And within this last stage, there is a difference between pupariation and pupation, but in order to make that distinction, I would have had to look at the contents, and I couldn't do that for my study. So for simplicity's sake, I'm only going to refer to this last stage as the pupal stage, and individuals as pupa or pupae for plural. So there are several factors that can impact blowflies and microbes on carrion. I chose to focus on 2, temperature and predation. And this is primarily because in many parts of the world, including Texas, temperatures, specifically ambient temperatures, can reach up to 40 degrees Celsius, if not higher. However, on carrion, temperatures can be just as hot, if not hotter. So as you can see here, it's a thermal image of a pig carcass placed out in College Station during the summer. And temperatures can range anywhere from 30.7 degrees Celsius up until 60 degrees Celsius. Now, blowfly larvae can thermoregulate by moving to cooler areas of the body, but they also risk being exposed to predators. But predation isn't 100% successful all the time. And so unsuccessful predation attempts can lead to larvae being injured, and also microbes being introduced in the process, which can lead to infections later on. So understanding how factors impact these organisms can allow us to have a better understanding of carrion ecology, but can also have forensic applications like in forensic entomology, which is the scientific study of insects and related arthropods associated with legal proceedings within a judicial system. So in a death investigation, forensic entomologists can provide an estimate of the post-colonization interval, which can be used to infer the minimum elapsed time since death. But to do this, they need to first estimate the age of insect specimens based on either the size of immatures or developmental progress using reference data. But the problem with reference data is that it's collected in the lab under relatively benign conditions. But in nature, there are several factors that can impact size and developmental progress of these insects. So for any life history trait, there's usually a distribution in size or developmental time, depending on the species or the trait that you're looking at. And my project aimed to look at whether heat shock and injury could cause a shift in this distribution. So specifically, I'm looking at the impact of heat shock and injury on life history traits of Cochliomyia macellaria. So to do this, I first wounded larvae using an aseptic wound or septic wound, placed them in the incubator for 24 hours to allow them to recover or for any infections to develop, then gave them a heat shock, and then put them back into the incubator and monitored them until eclosion. So I'm only going to be showing the results for some of the life history traits that I recorded just for the purposes of time. So here's a figure showing survival following injury. On the x-axis, you have treatment, and the y-axis, you have percentage of survival. As you can see, survival was lowest for septic wound. However, analyses showed that wound type didn't significantly affect survival after injury. For survival to eclosion, in this figure, the x-axis again is treatment, and the y-axis is the mean percentage of survival. If you look at heat shock treatments compared to similar treatments without heat shocks, as indicated by these boxes, the treatments that received heat shocks were always lower. I also looked at pupil weight, and so for this figure, the x-axis is treatment, and the y-axis is weight in milligrams. So all treatments were significantly lower than the control, as indicated by the asterisks. And if you compare heat shock treatments with similar treatments that didn't receive heat shock, as indicated by these boxes, comparison between these treatments showed that heat shock treatments would always have a lower pupil weight, and that difference was significantly different. So how does heat shock and predation potentially impact carrion decomposition? So we know that insects and bacteria, or microbes in general, are important players in carrion decomposition. And this importance is highlighted by a study conducted by Peckel et al. So on top here, you have different stages of carrion decomposition indicated by different colors. On the x-axis, you have time in days, and the y-axis is carcass biomass. So when bacteria and insects are involved in carrion decomposition, under the environmental conditions of the experiment, pig carcasses placed in the field by day 8, right here, were in the dry stage, and a good amount of the biomass was removed. However, when insects are excluded from the pig carcass, you can see that both the fresh and the bloat stage were extended, and not as much biomass was removed. Lastly, if insects are initially excluded from pig carcasses and allowed to colonize later on, the pig carcass did reach the dry stage at day 8, and again, quite a bit of biomass was removed. So heat shock and predation can immobilize larvae, and in severe cases can result in death. So if a majority, if not all of the larvae are immobilized, then decomposition could be delayed or slowed down. And in severe cases where all the larvae are dead, either due to heat shock or high predation success, then you might have a situation similar to the second scenario where you only have bacteria on the remains. However, if all the larvae are dead, but a new generation of insects or blowflies are allowed to colonize the body, then you may have a situation similar to the one in the bottom here. Heat shock can also impact microbes. So if it gets too hot and the microbes are also dying on top of larvae dying and there not being any insects, then you may have mummification results, and you might just have a bunch of dead things in the environment for longer. So in terms of implications in forensic entomology, heat shock and injury both can impact the life history traits commonly used as indicators of insect specimen age. So forensic entomologists should take these into consideration when estimating the age of insect specimens if there's evidence that heat shock and injury occurred. So in summary, heat shock and injury both can impact the life history traits of Cochliomyia macellaria, but heat shock had a greater impact than injury. Blowfly larvae and microbes are important players in carrion decomposition, and their survival and presence can impact the amount of biomass removed and the rate of decomposition. If blowfly larvae and other insects and microbes are removed from the remains either due to heat shock or predation and resulting in death, this could lead to the mummification of the remains. And lastly, there is evidence that there is a shift in distribution for life history traits examined. So because heat shock and injury do impact these life history traits, forensic entomologists should take these into account when estimating the age of insect specimens and if there's evidence that heat shock and injury occurred. So I'd like to thank my committee, Dr. Erin Cherone, Dr. Jeff Tomerlin, and Dr. Tony Kreppen, my lab mates in both the Cherone lab and Flies facility, and Dr. Woolley for letting me use his microscope, which I use to measure some of the life history traits. So I have some references, and with that, I will take any questions you may have.
Great, thank you, Jeffrey. If anyone has any questions, you can either unmute your mic and ask, or you can post it in the chat. So I guess I do have a question. So when you injure your maggots, you're trying to replicate what they could see in the wild. So what are the limitations to that? So what else could you potentially, like, what's different in the wild? So if a fire ant does it, they'll also inject venom. So that's inherently different, but are there any other types that are very similar to that type of injury that you know of?
Yeah, so for wounding, we used insect pins, and that was to mimic the mouth hooks of Chrysomya rufifacies, especially the larvae. So different bite sizes or could affect how severely they're injured. And as you mentioned, fire ants could also inject chemicals that may make the situation worse. So
I guess this could be a question more for Constance. Do you know of any maggots that survive a fire ant sting? Or
so we, not that I know of. So we did try to do some preliminary stuff because we were going to try to introduce organisms into it and not just do a lab base. But just from like the few larvae that we injured, it didn't seem like it. In fact, it was really hard to take them out. So, and if you drop it on, like if you drop larvae on a fire mound, they're more or less going to just not make it. Yeah. But I haven't seen any studies specifically looking at fire ants though.
Okay. Okay, and if anyone has any questions for Jeffrey, if you're watching online or you have questions later, you can find his email on our program book through our website, and you can feel free to email him with any questions that you may have. Thank you, Jeffrey.
Thanks.
So we will wait 5 minutes, and then Casey will be presenting. So I will be making Casey host soon, and then she'll share her screen. Okay. So for our next speaker, we have Casey Flint, and she's going to be presenting Small But Mighty, Microbes Influencing Decomposition and Attraction of Cochliomyia macellaria.
All right, thank you, Sam. So before I begin, I know this whole session is about decomposition. So if you have not, or I guess if you have been watching all these presentations, some of the information might get a little repetitive when we talk about what decomposition is. And then I also do have a warning that there are some decomposition photos in my presentation. So if that makes your stomach uneasy, please be aware of that. Okay. So there are 6 widely accepted stages of decomposition first described in 1965. And so these are shown in the photos on the right side of the slide. The fresh stage is labeled as photo number 1. And this is immediately after death when cell autolysis begins. And the organs affected first are the ones with high enzymatic activity. And the microbes within the body begin to flourish unregulated. And that leads into the bloat stage, which is photo number 2. So you can see that the legs are kind of spread out, and it's classified by bloating and putrefaction and the presence of necrophagous insects. This bloating is mostly in the abdominal region because that's where a lot of our microbiome is. It's in our gut. And so when the fluids begin to leak out, they'll leak out of natural bodily openings, and sometimes the skin will actually rip. And at this point is when insects will start to colonize, and there will be heavy insect presence that will lead into the 3rd stage, which is active decay. And in this stage, there's heavy insect activity and a dramatic mass loss. And depending on whether the duration of the stage can vary significantly. And then when we get into advanced decay and dry remains, there's basically less soft tissue present, and then finally only skeletal remains are present. And so when we look at this from an entomological standpoint, there's actually a lot more than insects that are present in decomposition. So we're actually looking at a lot of different disciplines interacting with each other. So there's the cell biology through autolysis and cellular level decomposition. There's microbiology through the microbial metabolism of the tissue and proteins, as well as entomology with the insects consuming and utilizing the remains, as well as several other disciplines. But now we want to focus kind of on the entomological stages. And a common way to determine a post-mortem interval estimate is to use a time of colonization estimation based on the insect evidence present. And so this time of colonization interval is based on the duration of the post-colonization interval shown in the red box here. And this is calculated based on temperature data and development studies. And overall, this is a widely accepted method for time of colonization intervals. But this pre-colonization interval has traditionally been neglected. And so we're not exactly sure what is dictating the length of this interval, whether it's microbes or flies or time of day. There's a lot of different things that can impact this. But one of the consistent things throughout all the studies conducted in forensic entomology is the microbiome, right? But what happens when one of these factors are removed? And so first we're going to look at the impact that insects have on decomposition. And so insects will significantly increase the rate of decomposition and tissue removal. So if you look at the photo on the left, the top graph is the mass left during each of these days. So you can see that in the first 5 days, there is a significant decrease in tissue present. So about 90% of the tissue was removed in the first 5 days. And the bottom graph is showing you the mass present when insects are excluded. So there's a vast difference in these graphs and how fast this tissue is being removed. And so one way that the insects impact this, besides the mass loss, is they actually alter the microbial community. And a lot of this is through antimicrobial peptides that these flies produce. And they produce these to suppress microbes in order to reduce competition. And a well-known case of this is Lucilia sericata, because they're used in maggot debridement therapy. They consume the necrotic tissue and they produce these AMPs to promote healthy wound healing. And so we know that these blowflies manipulate the microbial community and succession trajectory. And so if you look at these 6 photos, on the left we have 3 pigs that were accessible to remains immediately, or accessible to insects immediately after placement in the field. All of these little white specks that you see in the photos are larvae that are feeding on the remains in the soft tissue. But the 3 pigs on the right were placed exactly at the same time as the pigs on the left, but they were placed inside insect exclusion cages. So there's a difference in decomposition rate here. So on the left, they're in active or advanced decay or decomposition, and then on the right, they are still in the bloat stage. And so they are being consumed from the inside out by the microbes present. So when we talk about microbes, what do we really mean? So when we look at the human microbiome, we are actually outnumbered human cells 10 to 1. So there are 10 times the amount of microbial cells to human cells when we look at our body as a whole. And so throughout our body, there is high variation based on like location in our body. So in the top left graph, the oral microbiome is clearly different than the skin microbiome, which is different from the gastrointestinal microbiome. So everywhere we look in our body, there's a unique composition of microbes. And so when we die, these microbes flourish after death. They spread throughout our body on a somewhat known succession trajectory, which we'll discuss a little bit on the next slide. And these microbes will produce volatile organic compounds that will cause the bloat stage and attract insects, right? But everyone doesn't have the same microbiome, right? It's impacted by our immune system, who we're with in our environment, our lifestyle, our diet. So everyone really does have a different microbiome. But is it different enough to impact decomposition? And so this is thought to, there's a new concept called a microbial clock. And this is done with mouse studies. But what we've done here is we've essentially bottlenecked their microbiome due to the colony care protocols. So all these mice tested have a very similar microbiome. But we can see that throughout decomposition, there are significant changes in their community structure that we can essentially use to determine a time of death, right? But these estimations would only be accurate until insects colonize a carrion resource, because the flies that colonize will then alter that microbiome. But how do we know when this happens and what that succession trajectory would look like before the flies colonize? And so this is a study that was done to where they looked at insects, the impact of insects at different times of colonization. So the top row is immediate colonization, the middle row is no colonization, and the bottom row is delayed colonization. So we can see that insects have a significant impact on decomposition, right? But what happens when microbes are excluded? And so this led us to have a study with exenic and non-exenic mice. Exenic mice, it is a mouse that has absolutely no microbes in it. So it has no microbiome. And then we have non-exenic mice, which have the standard lab mouse microbiome. And so what I did was I placed them in mason jars, covered it with parafilm, and then I have a charcoal filter to filter the volatile organic compounds that were coming in and out. And then I also have a 0.2 micron filter up top, and that's basically just to ensure that these axenic mice stay axenic. And I just simply let them decompose. And I took pictures every day so I could tell the difference in the decomposition stages. So I know this slide is a little busy, but what we can see on the top is the axenic mice. So these have no microbiome. And what we kind of see is I want you to focus on the abdominal region of these mice because that's where most of the bloating occurs. And if you look at the axenic mouse, you can kind of see that it just almost caves in on itself. In the non-axenic mouse, there is this bloating stage present. So over the first 6 days, there's this clear difference where there's either a bloat stage of the decomposition process or there's no bloat stage. And these photos show it a little more clear, the axenic mouse. If you look at the day 6 photo, you can see the indentation, and it's just kind of folding in on itself, where the non-axenic mouse, the feet are still up in the air, they're separated from that abdominal bloat. And so what this really gets at is decomposition really decomposition with no microbes? And so without microbes, it's just the enzymes that are working to break down the cellular components. And this is really more of a decay because there's no external consumers of the remains. Whereas when we have the microbes present, it's more of decomposition because those microbes are separating and breaking down the tissue into its components. But in reality, how does this affect blowfly attraction and colonization? So I use Cochliomyia macellaria. It's a very common colonizer of carrion in the United States and especially Texas. And it has a wide variety of applications. So I swabbed the mice to begin with, plated on TSA plates and blood plates. And then I put them in sterile mason jars. And you also used a 0.2 micron filter. And I used a dual choice cube olfactometer. And so the arms were set up like so with sticky traps to trap the flies that went into the elbow, and then the treatments were in the mason jars at the end. And the parameters that I used were from preliminary studies. So I ran 3 different treatment combinations to make sure that none of the treatments were actually repelling the flies. And then I ran this over 8 days. And so at the conclusion of each cube, I sexed and determined the ovarian status of the females. And then once again, at the end, I plated to ensure that the axenic mice remained axenic throughout the experiment. So all in all, the treatments were not repellent. So this is what happens when we put the axenic mice against the non-axenic mice. And in the first few days, there's not really a huge difference, but when we get to day 4, that's when the non-axenic mice are really attractive to these flies. And so when I finally break that down by sex, the non-gravid females and the males, their response patterns look very similar to this graph. But what's interesting is that the gravid females essentially never found the axenic mouse more attractive than the non-axenic. There's no real overlap of these means here. And so it's very interesting when we look at these results, because what we can tell is that these microbes are they're really the ones driving the attraction of Cochliomyia macellaria. And there's also this temporal variation because you can see that the patterns are changing over time. And so when we look at the variation between the microbiome of different individuals, it could potentially affect the attraction of insects post-mortem. And this will get into the pre-colonization interval variation that we look at in forensic entomology, which could theoretically impact a post-mortem interval estimate. And so in the future, we are wanting to look at the thermal impact of these microbes within the carrion resource and impact on the attraction. So we would rerun this experiment at different temperatures. And then another thing that we want to do is take the axenic mice and inoculate them with known microbes. We would use some of these present in the graph on the right, just because they are commonly found in decomposition. And so we'd like to isolate them and see what they what they do inside of the carrion remains, or the carrion resource, I'm sorry. And so with that, I'd like to acknowledge my committee members, all of my Flies facility lab mates, Texas A&M University, and my husband for putting up with all of the stinky smells that go along with my research. And with that, I'll take any questions.
Thank you, Casey. If anyone has any questions, they can either unmute their mic and ask the question, or they can ask it in the chat, and I can read the question from the chat. And we have a couple minutes, so if people are typing, we can wait.
Yeah, sorry, I know I was talking kind of fast.
No, that's okay. Just to give everyone a minute, if anyone has any questions for Casey and they're watching this after the fact and watching the recording, or they come up with a question later and really want to ask, you can contact Casey through her email, which is on our program book that you can locate through our website. All right, and I'll keep the questions up and I'll start sharing my screen.
Okay, let me.
You can see my screen all right, correct?
Yes.
Okay. All right, again, if you have any questions for Casey, feel free to email her, which you can access through our program book. Okay, it's 11 o'clock. So I'm actually the last speaker, so I'm going to awkwardly introduce myself. My name is Samantha Sawyer, and I'm presenting Vultures, Ants, and Flies, oh my, competition for small carrion between scavengers across habitat, season, and carcass type. So we're in the Carrion Ecology section, so you've probably heard multiple times about carcasses. So you know that there's going to be some photos of carcasses in this presentation. But carrion is considered an ephemeral resource. So this means that it's short-lived in the environment. And because of this, it facilitates this really intense competition, particularly between three major groups. So as Casey had talked about, microbes, and as well as invertebrates. So these other talks that we've had during this session have been mostly between competition between other vertebrates, or in Casey's case, vertebrates versus microbes. In addition, we also have vertebrate scavengers that will consume these carcasses rather quickly. But in addition, though they're not competing for these resources, plants actually benefit from these plant resources quite a bit, as you can see in this photo. But for the purposes of this talk, I'm going to focus on the invertebrate community and vertebrates consuming these small carcasses. And the competition for carrion is actually influenced by the carrion itself, so different factors surrounding the carrion: what carcass is it, what species is the carcass from, and the general biomass, as well as these abiotic factors, so environmental factors surrounding this carcass will drive this competition. But we know that these small carcasses, particularly less than 10 kilograms, have this really intense competition for it, because it's pretty easy to remove in the environment. And like I said, these resources are already ephemeral. So for vertebrates, we have this really fast consumption because these small carcasses are high energy resources and they're easy to get. So there's little risk associated with it, assuming that there's no disease involved with this animal's death. So they can actually grab the resource and remove it from the environment or consume it very quickly at the site. The only constraint they have is the presence of microbes. These microbes can produce these toxic compounds that will make it so when they consume them, they'll get sick. You can think of it, if you ate a rotting piece of meat, you would probably get sick too. So to avoid this, they do this early arrival where they will locate a carcass rather quickly, and they can do this through smell and sight, or they'll actually have these physiological adaptations to fight off these microbes. An example of this is vultures can have a pH of around 1 to kill bacteria such as anthrax. But the vertebrate community is largely facultative feeders, and this means that basically they're not relying on these carcasses for their main source of food. They just come across it and they eat it. So the diversity and the abundance of these animals that could consume these carcasses is vastly abundant in our environment. And we know that while these systems are understudied, that they release these nutrients into upper levels of the food chain, meaning that they can have top-down cascades on the environment. When we look at our invertebrates, most of the individuals that colonize these carcasses are using it for reproduction, specifically our flies. And what I'm going to focus on today is blowflies in particular. And a single jackrabbit, so this really small carcass, can actually produce over 1,400 fly larvae. And that's significant because when these individuals are feeding on these carcasses, they're introducing these nutrients into lower levels, so they're making these nutrients accessible to microbes and plants. But they also have these major ecological services that they provide. So a lot of people think of blowflies as pests, where they're transmitting pathogens, and that's true, or that they have medical importance because of myiasis, where they feed on a living individual. That's also true. But we tend to forget that these individuals are major pollinators. So they pollinate agricultural crops as well as our flowers. And this is vastly understudied and underestimated. So when we look at these two groups, we usually study them in isolation from each other. But in reality, we know it looks something like this, where it can have a vast diversity of individuals from both groups interacting with one another and competing for these resources. So one of the only studies that they have looking at vertebrate scavengers and insects is the Parmenter and McMahon study. And what they did in this study is had insect access only cages and vertebrate scavengers and insect access. And they found that decomposition rates were exceedingly higher with scavenger and insect access than insect access alone. But they didn't really look at what insects were present, but beyond what the presence-absence data. So they don't know the production of these flies. So the purpose of my study is to determine what habitat, season, and carcass type provides the greatest production of these flies. Like I said, these jackrabbits can produce a lot of different flies, but when we introduce a vertebrate scavenger into this, what effects does this have? So to study this, we had 2 sites, Coulter Airfield in Bryan, Texas, and the Texas A&M Farm. And we had open habitats and forested habitats indicated here. This is what the sites look like on the ground. So in the open habitats, it's always cut grass, so you can easily see these carcasses. And in the wooded habitats, you have these low grass areas with some tree cover. In the field, we had 12 carcasses in every habitat, and there's 4 of each carcass type. So we had broiler chicks, brown rats, and white rats, and we placed them 50 meters apart. And this is to prevent ground-dwelling vertebrates from moving in between carcasses and feeding on all of them in one foraging event. We also had 3 game cameras, 1 at each carcass type, to just get a sample of what type of vertebrates were consuming these carcasses. And we also had 1 data logger to look at the temperature at each habitat. And this is because insect development relies heavily on temperature. So as I mentioned, invertebrates are using these carcasses for reproduction. So between the time they're laid on the carcass until they're feeding third instar, they rely on the carcass to be in the habitat to survive. If the carcass is eaten while they are consuming the carcass, they will die. And if the carcass is removed from the environment, they won't be able to lay their eggs on the carcass at all. So we wanted to optimize this carcass utilization period. So we could estimate when we could pick up this time based on temperature. So we could do this through accumulated degree days. This is a way in forensic entomology we can predict development of insect flies and backtrack time. So I actually reversed this and projected when we would pick this up in the future. So in the summer, we used Cochliomyia macellaria. This is a studied species that has been in all of the previous studies, and this is because this is a really relevant fly in our area. So I used the Boatwright and Tomberland development set for that and calculated it to be about 3 days based on the temperature during July and August of 2018 and 2019. And we also did this in winter. We did this in December 2018 and 2019 as well. But we used Formia regina, and this is because Formia regina is a common winter species where Cochliomyia macellaria tends to not be as abundant. So we calculated this to be about 60 some odd days. But after 8 days or within 8 days, all of these carcasses were removed. So that's when we concluded the study. And we used the Nunez and Vasquez data set to conclude this. So every day for the duration of the study, we checked the carcasses at dawn and dusk to look for a couple different things. So we wanted to see if there was insect activity and what carcasses were taken. Any of the carcasses that were left at the end of the designated time period were brought back to the lab by being put in these rearing chambers. And then we reared them out and identified them all to species. So all of our statistics is through R, and we looked at ANOVA and Fisher's LSD to determine what site, habitat, season, and carcass type was the best for different types of carcass bait. When we look at what animals are present at our game cameras, we can see that there's quite a diversity. So we had individuals such as armadillos that aren't consuming the carcasses necessarily, but they're feeding on the insects that are at these carcasses. So they're disturbing the carcass in some way. And then you have the non-surprising scavengers, such as vultures, foxes, possums, skunks, and coyotes. And these are all mesocarnivores, so they thrive on anthropogenic activity. And just to remind you, we had a farm and an airfield. And this was surrounding College Station, so this is a major city. So these are expected. When we look at the different rates of scavenging, I'm just going to grab my laser pointer here. When we look at winter, both open and wooded habitats had 100% scavenging. As I mentioned, all the carcasses were taken before that development period. So that's really important because these flies aren't being able to utilize these carcasses in the winter months. But when we look at summer, there is a difference between open and wooded habitats, where in wooded habitats, these conspicuous colors, carcasses, so white rats and chicks, things that are really obvious in the environment, are being selected at a higher rate than these brown carcasses, which is probably more realistic in what you would see in the habitat anyway. And then you have these open carcasses that generally aren't selected for more so than others, but these chicks tend to be, and this is a highly variable, but they tend to be selected at a slightly higher rate. But again, this is a very variable treatment. So when we look at scavenging, there's been studies that look at season. So we know that in the summer, there are more things or less carcasses scavenged than in the winter. And this is thought to be because of the microbial community, so that early access being really important. But in our study, we saw anywhere from 53 to 91% scavenged even in the summer. And in the winter, we saw double what this previous study had seen. So the question is why is this so high? And that could be because of anthropogenic land use, which can actually impact the distribution and ecology of these animals. Particularly, they found that fragmented landscapes like the sites that we have negate temperature effects. And they also saw in these fragmented habitats that even in the summer months, they saw up to 96% scavenging. So this makes sense. And they think this is because of trophic downgrading. So when you remove this apex predator from the environment, you have these mesocarnivores increase in abundance, and they're more likely to scavenge than other animals in the environment. But we also have this open versus wooded habitat. So we know that open habitats allow for site-reliant scavenging, particularly from our vultures. But you also have this high contrast in fur color in wooded habitats that we see. And the animals that are consuming these carcasses are different than these vultures. So it could just be difference in their biology. When we look at the flies that are present, by and large, it was Lucilia exemia that we found at the carcasses. But there were a couple that we did find Chrysomya rufifacies. When we look at the utilization of flies for these carcasses, you see that brown rats tend to have a higher colonization rate than these other carcasses. And again, in winter, there was no utilization by any flies. But when we look at open habitats, rats are used and chicks aren't at all. But if you remember earlier, the vertebrates didn't consume all the chicks. So the question is, what's going on here? And so we actually had a really high prevalence of fire ants. And this wasn't the main study goal of our study. Constance Lynn actually is studying the competition between these two individuals, but they did have a significant impact. Okay. So we know a couple things about our fire ants. So we know that they can remove maggots from the carcass. We also know that they can bury carcasses, so they can take things from the environment and cover the carcass, and this prevents other flies from colonizing these carcasses. In addition, they can just remove the biomass from the environment completely. So when we look at utilization from fire ants of these carcasses, you see that chicks are used quite a bit in these open habitats, but these other carcasses are used mostly brown rats in the wooded habitats. So flies aren't even using these wooded habitats as a refuge, utilizing these brown rats. So we wanted to take this a step further, and again, this is something that Constance is looking into much further. But we found that in carcasses without RIFA and carcasses with RIFA had a significantly different production of flies. So we see that when you don't have fire ants present, you have this significantly increase. And this was significant for presence and habitat, which isn't surprising because fire ants were seen mostly in open habitats. But when we look at species richness, Chrysomya rufifacies was only found at white rats with RIFA, and also these were in open habitats. So there's something interesting going on here. So the question was, what provides the best production for our flies? And we found that wooded habitats in the summer and brown rats are the best producers. But we already could estimate this, but this is even limited because only 30% of the carcasses were consumed by flies, and that doesn't even include if they had fire ants, which limits their production significantly. So what does this all mean? And I'm almost out of time, so I'm gonna go through this pretty quick. So blowflies are under a lot of selective pressures in the habitat as is. So they have other things competing with them, and vertebrates are really good at locating and consuming these carcasses before they can develop. Habitat fragmentation is driving vertebrate scavenging to increase. So these anthropogenic factors are putting selective pressures even higher for these blowflies. And in addition to all of this, invasive species are also altering their availability of these carcasses. So Luciliexemia and Chrysomyia rufifacies are native to South America, where these fire ants are found, but they're still having an effect on them. And this can have long-spanning ecosystem effects. So when you're removing these carcasses, the flies are actually probably locating things for anthropogenic wastes. So things like our trash, they might be utilizing those more so than carcasses in the environment because these vertebrate scavengers are consuming them more so than they are. And on top of that, fire ants are consuming them as well. So I don't have too much time to go on all this, but we are doing a slew of different research with Luciliexemia, and I suggest going to check out my undergrad's posters. Luciliexemia is a vastly understudied species, so we're doing basic biology to study those and looking to see if we can have selection for specific carcass types in these flies. With that, I am a little over. I want to acknowledge all of the research assistants I've had in the field. I was very pregnant when I was doing this, so I really needed a lot of help. So David, Lauren, and Sophia, thank you so much. And also my husband, who, when they weren't available, went out with me. My trail cameras were also from our plenary speaker, Dr. James Beasley. So those same cameras were used in Chernobyl, which is pretty cool. And support from Fly's facility. And with that, I will take any questions. And you can ask questions either through your microphone or in the chat. I can wait a minute if people are typing. So Zante asks, could the effect of feathers versus fur be impacting on what you're seeing in chicks versus rats? Potentially. I know that the broiler chicks, so they do have feathers, but they're not like down feathers, they're very fluffy. And when we see colonization, particularly of Luciliexemia, and this was in rats and chicks, they were colonizing underneath the body, not utilizing the orifices. And this could be for temperature reasons. In the summer, it was getting really hot, but it could also be from fire ants as well. They're using underneath the body to try and get away from them. But we also know that fire ants do bury under the body too. So it could be, but it's a little hard to tease that out. It might even be more so from biomass than from the type of like fur or feather. All right, and if anyone does have any questions for me, no problem, Zante. If anyone does have any questions for me, my Twitter is right here. I also have my ResearchGate up and my personal email. So if you're watching this on recording, you can feel free to ask me questions on any of those platforms. And that concludes our, let me stop sharing my screen. Okay. And that concludes our session for Carrion Ecology. So thank you all for watching. We do have sessions later on in the day, so you can check out our program book to look at specific titles. But also after the lunch period, starting at 1 o'clock, we also have a bunch of posters in our poster room. So you can go check that out as well. And the award ceremony for all of the presenters is at the end of the day. The link is available on our website. So thank you.